let's dive into the article because that's what Journal Club is all about. And so, were you guys able to get a chance to read the article and do you guys have initial thoughts about it? What did you think of it?
It brought back a lot of stuff from biology and reminded me of stuff I realized I'd forgotten that we learned. So it was a nice refresher in that. And um, yeah, it was interesting to read.
Yes, definitely. Yes. Whenever I read mitochondria, I was like, it's time to think back to ninth grade biology. Okay, Mackenzie, what did you think about it?
I thought it was super interesting. Definitely similar to the ones we read this summer.
Mhm.
hmm Yeah,
Yeah.
brought back a lot of high school biology for sure.
Yeah, I must say, whenever I saw the art, like the title of the article, I was like, what is that talking about? But I think that the article did a really good job of talking about, like different mutations and the complexes, as well as like what is AML. Because I had no idea about AML, but it's like a very scary type of cancer. So I found that to be very interesting. Yeah, so this slide like talks a little bit about like the authors. It was published in 2018, so it's like relatively recent. And it was also published in the Nature Journal, which is very high impact. It's very impressive. And so, yes, so here are a few discussion questions I came up with. So, would one of you like to give a little summary about the article, like what was it about, or like what topic was about?
I can. So, at least from what I interpreted from it, was that during the electron transport chain, that whole cycle, there can be errors and mutations in the mitochondria, and that can affect like the overall survival rate in patients with acute myeloid leukemia.
Yes, definitely. I was amazed because I didn't even know that there could be mutations in the electron transport chain complexes, but it's crazy how just like a few little changes in the mitochondrial DNA can result in such damage. See, I made like notes on this. I went a little bit crazy, but yeah, so are you guys familiar with what AML is? I did a little bit of background research before I started because I didn't know.
Not much more than it's a kind of leukemia, so I'd love to hear what you found out.
Yeah,
Yeah,
same.
sure. So, yes, so what I found out was apparently it's a type of cancer that occurs in like blood and the marrow region of the body where white blood cells and red blood cells are formed. And so what happens is that the body produces abnormal white blood cells. But what happens is that like because of the abnormal white blood cells that's produced, then that results in the body not being able to, beneficially fight off infections, as well as it like prevents the body from getting adequate supply of oxygen. And so that's what I found. The white blood cells, they end up like growing very abnormally, very quickly. And so they're not able to develop properly, and therefore they're pretty dysfunctional. So I had no idea, but that's kind of crazy. Why do you guys think that like the topic is important? Like why do you think that the researchers decided to focus specifically on like mitochondrial DNA mutations and with regards to like the ETC?
I know at several points they were saying points for future study, and I think they said something about it's useful for um, like future medicine production and biomarkers. So as they're getting more specific on what causes the cancer, they can uh, figure out how to target it and how to uh, improve treatment.
Mhm. That's a good point, yes. Yes, cancer is just a crazy disease, and that's why it's like really important to like look at all of these different biomarkers and see, well, these are the mutations, this is what we've found out, and so how can we use that in order to develop treatments? And so here's figure one. What are your initial thoughts? What do you think this is saying? I can give you a few, a minute or so you can read the caption as well.
One thing I wasn't sure about, one question I had was about the TP53 mutations. Are they mutations in the mitochondrial DNA or in like the nuclear DNA?
It's a very good question because I know that the study it mentioned how like there was a significant association between mutations that happened in the ETC as well as like TP53 mutations and how it like further worsened the prognosis. Let me see if I can go to the article and find it.
Yeah, I was looking in the article. I think it might actually be the mitochondrial DNA and like the second paragraph down because it's talking about how mutations in the mitochondrial DNA have been identified in malignancies.
Oh, okay. You're talking about this paragraph?
Yeah, like right on the
Oh,
bottom.
okay. Interesting. Yeah, I also found it very interesting how it didn't mention the TP53 mutation in the introduction.
Yeah, that's what I was looking for and I couldn't find
Yeah.
talking about. Weird.
Yeah, because I feel like articles that normally like would introduce everything that they're gonna find in it, but I didn't see that. Maybe that's like a suggestion we can have, an improvement. Oh, okay. So it's related to P53, for making the protein. So I guess it is a nuclear DNA, like it's related to that. It's very interesting. Okay, so TP53 is a suppressor, excuse me. So it makes a protein, tumor protein P53, which acts like a tumor suppressor. So I suppose whenever it's mutated, that can end up causing the mitochondrial DNA mutation and resulting in the cancer. Sounds good. Did that help with your question? I know it was.
I think it did, but
Oops.
now I'm okay because if this is a like a nuclear DNA thing, and then they're saying it relates to the mitochondrial DNA. I think that's interesting how you can be more likely to genetically develop a mutation. I think it said something about that.
Mhm.
I highlighted it because I didn't know what it meant at first. Um, molecular risk. I thought that was interesting, like the probability of developing a mutation.
Yeah, I wonder how the mutation, like how mutations in the mitochondrial DNA happen. Like I know DNA mutations can happen with DNA polymerase, but I've never heard mutations in mitochondrial DNA. Very interesting. Okay, so what do we see that this figure is saying?
I think is it just talking about how it's like statistically significant the number of patients with AML that also have mutations in the mitochondrial DNA.
Hmm. Yes. Right, because these p-values, they denote significance.
That's interesting too that they're looking at like the statistically significant difference in patients with the TP53 mutation between patients with mitochondrial mutations.
Hmm.
So it's like you can have the TP53 mutation without mutations in the mitochondria, but
Yeah.
still they coincide.
Mhm. Yeah, I found it interesting how it's saying like that there's a higher percent of patients with ETC mutations that also have TP53 mutations. And I also found the chart like very interesting. I've never seen a chart kind of like that, how it's like stacked like that, but I think it's very interesting how it's like talking about the percent of patients and it all adds up to 100%. So it's a very interesting looking chart as well.
Um, I see. So the blue is the mutated TP53. Is the WT, does that stand for wild type, as in not mutated?
Yes.
Okay.
Yeah, so like the yellow is signifying how the TP53 in those patients are perfectly fine, no mutations. And then the blue is saying that it's mutated. Okay, so are we ready to look at the second figure? I think this figure was very interesting personally. I'm excited to see what you guys think about it as well.
No, I thought it was really interesting how it changed overall survival rate, but not disease-free survival. Because I guess once you like fix the issue, like their chances at survival are like similar enough that there's no statistical significance, but like
Mhm.
without treatment or like without the correct treatment, there is a difference.
Mhm. Yes, I agree with you. Yeah, whenever I saw like this, like I don't really know how to say it, like Kaplan Meyer, something like that. I wasn't familiar with what it is, so I searched it up and apparently it's widely used in medical research and it shows how like different, like different treatments or different conditions can impact the survival. So I thought that was pretty cool. What do we think these graphs are saying?
Yeah, I think it's just saying that the mutation does make a significant difference in their chance at overall survival.
Like Mackenzie, you're spot on when you say that like patients with the mutations in the ETC showed significantly lower overall survival compared to those without mutations. So I guess that's the main purpose of this figure. Okay, we can move on to figure three. That's good with everyone? Okay. Yeah, so figure three, it looks very similar to the previous figure. Were you guys able to look a little bit at it, digest what it's trying to say? Okay, so what do you guys think it's saying? And what do you think is like the difference between this figure and then the previous figure that we looked in?
I think this one is stratifying it by complexes, and it's interesting because it makes it seem like um, no patients with mutations in complex 1 or complex 3 survived even past 50 months. Like they barely made it to 25 months,
Hmm.
which is interesting. So that shows the uh, fatality of those mutations.
Hmm.
Yeah, and I would say what makes this one different than the one before is that they noticed there was no statistically significant difference in the disease-free survival, so they focused all of them on the overall survival and then got more into detail about comparing the different complexes.
Hmm. That's a good point. Yeah, and I was just amazed at how mutations in like specific complexes can determine how well you survive rather than or in comparison to other mutations. So I guess it's very specific what this research did. It was like I'm gonna figure out which complexes the mutations are worse, see what prognosis is worse, and base the research on that. So that was very interesting. And yeah, as Emma said, they found that mutations in complex 1 and complex 3 were worse. Well, resulted in less percent of people surviving when compared to complexes 4 and complexes 1 and 4. So we can go on to figure 4. Are you guys ready to discuss a little bit about this? Okay.
I think it's interesting how none of the patients without transplants, none of the uh, patients with mutations without transplant survived to the 50 months. And it's also interesting, it looks like even with the transplant, they still had a lower survival rate, even with the transplant, which I think is interesting.
Hmm. Yeah, why do you think that is? Do you think it could have been like transplant rejection or something? Because I would think if someone gets a transplant, then they would have, pretty good working like body parts and organs. But I was pretty interested in how they just didn't survive.
I wonder what kind of transplant it is too, since it's your white blood cells. Is it like a stem cell transplant then?
Well, I was assuming it was a bone marrow.
Oh, yeah, that would make sense.